Okay, so it's time to talk about communication of information. This chapter centers on these ideas. Um, these are called enduring understandings. These are major themes in life on Earth. That first, expression of genetic information involves cellular and molecular mechanisms. In other words, it's obvious, and you should have learned this in the last chapter, that DNA, that's genetic information, expresses itself through complicated chemical pathways. And part of that is just that transcription and translation mechanism we went through in the last unit. Secondly, the processing of genetic information is actually a source of gene variation in a population. Turns out that viruses don't always process themselves the exact same way in every cell, and as a result, they make mutants. Just it's part of the way they make their bodies as they're reproducing inside of other cells, and that helps them evolve chemical resistance to immune systems that are trying to kill them. Next, cells communicate by generating, transmitting, and receiving chemical signals. That's the hormone concept. And then lastly, living things actually communicate with each other through colors and sounds to tell other living things, do this or don't do that. So these are the ideas. I probably don't want to bore you with all the individuals, but we're going to be talking about all kinds of things in this unit. Ultimately, we start out at the level of the gene, which is these first things, and then we talk, and then how genes are regulated. Then next, we talk about how cells communicate with each other through signal transduction. And lastly, we talk about nervous systems and how living things communicate to one another. So first we start out at the level of the gene, then we move up to cells, then we talk about whole organisms. So let's scroll up a little bit so that we can get the next topic kind of a little higher on the screen. It turns out that there are four basic kinds of genes in animals. Before we dive in, just to let you know, there are, now these are different too, DNA regulatory sequences we're going to be talking about today. Those are things like promoters and operators and enhancers. Those actually are important in this chapter, as well as regulatory genes, which actually make regulatory proteins. Regulatory sequences don't make regulatory proteins, and you'll see in just a minute. Regulatory genes do make regulatory proteins that bind to regulatory sequences. And then lastly, remember those introns from the last chapter? They're actually also involved in regulating gene expression. So the gene activity that we have in our cells is being modulated and controlled by DNA regulatory sequences in our chromosomes that actually don't make any proteins, other DNA regulatory genes that do make proteins, which bind to regulatory sequences, and lastly, some of the intronic regulatory RNA that's made through transcription processing. Now, there are four kinds of basic genes in cells, and there's overlap here between these because homeotic genes are inducible or repressible at times. But basically speaking, there are homeotic or embryonic genes that only work when a living thing is embryologically developing from a fertilized egg. Those genes are different from housekeeping genes in that housekeeping genes are being made all the time. I mean, they're always active. Housekeeping genes make like the genes for glycolysis, the Krebs cycle, the electron transport system. They always got to work because if you run out of those proteins, you're dead. Homeotic genes only work, on the other hand, when you're an embryo, and they do not work anymore when you're an adult. So these two kinds of genes are kind of vastly different in how they act. The last two are genes that are not absolutely critical for everyday life, but we do have them. Inducible genes normally don't do anything until they're switched on by some chemical that comes in the cell. So normally they're switched off and then they get switched on by a chemical. Repressible genes are normally switched on, but then if some chemical disappears or something, they can be switched off, and then as a result, the cell doesn't use them. So there are four basic genes in a cell toolkit: embryonic genes, everyday housekeeping genes, and then these inducible and repressible genes, which they don't have to use all the time, but they will if they need to. Just so you know, before we dive in, you really don't need to transcribe and translate all the genes you have in your cells all the time, because it'll waste a lot of energy. For example, we don't make any homeotic proteins as an adult. If you do, that means you probably have cancer, and that's not a good thing. And then next, there are many enzymes that you don't need, unless the substrate's around for the enzyme to break down. Example, lactose. Lactose is only found in mammalian milk, so it's found in breast milk of pretty much all mammals, or there's probably some exceptions, but most mammals anyway. But adults don't normally drink breast milk except for humans, and so therefore don't need to make lactase. Lactase is the enzyme that breaks down lactose into glucose that will basically get broken down in glycolysis. So lactose is made of glucose and galactose, and those two chemicals can be processed by glycolysis. Lactose can't be processed by glycolysis. So the purpose of lactase is to break down lactose into stuff that the cell can use in glycolysis. Well, it turns out that most people don't make lactase unless they drink milk. If you're lactose intolerant, it means that you've lost the ability to make lactase. It's no longer inducible. So in most adults, lactase is an inducible gene, meaning it's switched off by default. You don't make lactase unless you drink milk and it switches on. The only thing is, in the human body, if you suddenly drink milk after not drinking it for a long time, you'll probably get digestive problems, and the reason for that is it takes a day or so to switch on to get a lot of lactase on board, and that might not be in time to digest the milk. And if lactose goes into your colon, the bacteria will go crazy, and you'll get all kinds of gas because they'll metabolize it, blah, blah, blah. And then probably you're going to have a bad digestive diarrheal event, and then after that, you'll be okay. Anyway, let's talk first about regulatory sequences, which are different than regulatory proteins. Regulatory sequences are DNA stretches that will interact with special proteins called regulatory proteins to control transcription. So regulatory sequences themselves don't make any proteins, but they're part of your DNA and they regulate what actual genes do. The most famous of these is the promoter. The promoter is a region of DNA just upstream from the TAC start codon of a gene, and it basically is used to control the transcription activity of the gene. The most common regulatory promoter in the human genome is the TATA box. The TATA box is a binding site for regulatory proteins to determine the rate of activity for RNA polymerase. So let's take a look at something you've already seen, but I need to remind you about. Let's go to gene expression. Let's go up here, right there. All right, now we saw this in the last chapter, and this was supposed to animate the transcription and translation of a gene. The little hand is RNA polymerase, okay? And what it did was it read the TAC, GCT, and so on, and it produced an RNA molecule, AUG-CGA, that's complementary. What a promoter is it's a section of DNA that is out here. It's upstream. It's, these are all nitrogen bases sticking up off the sugar phosphate backbone, about 25 base pairs upstream. That means in the 3' direction here. There's a TA nitrogen base sequence. That actually serves as a binding site for special proteins that can allow the hand, which is RNA polymerase, to either not bind or to bind more quickly to the TAC and transcribe. This box here is called the TATA box. So let's go back to the outline for communication and talk about that now for a second. Let's go there. So the TATA box is basically a binding site for what are called transcription factors. What transcription factors do is transcription factors are made by regulatory genes. They transcribe and translate transcription factors that bind to the TATA box to promote RNA polymerase to transcribe the gene that's downstream. Generally speaking, that's what the TATA box is for. The TATA box also, though, can be bound by histones. Histones are special structural proteins in chromosomes that help stabilize the chromosome and wind it up. And histones can actually bind to the TATA box and act as a switching off signal. So in general, the TATA box is not an on or an off signal directly. It depends what's bonded to it. It's a binding signal area. If a transcription factor is bound to the TATA box, generally speaking, you have enhanced transcription and translation activity or transcription activity by RNA polymerase. If a histone is bonded to the TATA box, generally speaking, the transcription level of the gene that's downstream is inhibited. In addition to the TATA box, promoter region, there's also a region called the enhancer. Unlike the TATA box, the enhancers are located like far away on the DNA, usually at least 100 base pairs or more. And they actually are even more effective at usually enhancing the level of DNA activity of a gene. And it's been discovered they do this because DNA actually in a chromosome is looped and looped and looped millions of times into bizarre conformations, and that brings regulatory sequences like an enhancer right there, right next to a TATA box on a gene that's right there. So if a regulatory protein binds to the enhancer and that attracts RNA polymerase here, the RNA polymerase will scoot downstream and transcribe and translate this gene that's located here at number 1. So this is complicated now. What we have here is a TATA box, which serves as a basic control region of this gene. The TATA box, if it's bound by a transcription factor, that will enhance RNA polymerase to come in and go and transcribe. If an enhancer regulatory protein is bonded here, that'll accelerate the transcription and translation even more than something bound at this promoter region. In addition, if a histone is stuck here, that will shut down all activity here, even if the enhancer is actually bound by a regulatory protein. One of the most famous regulatory protein motifs is this blue and gray ribbony antler set here. This is called the leucine zipper. The leucine zipper is a DNA binding protein that actually locks on to DNA. It's capable of binding at promoter regions and enhancing RNA polymerase activity. This thing serves as a flag. It attracts RNA polymerase in, so then it will go downstream and transcribe and translate a gene. So the leucine zipper is a common transcription factor that binds to human DNA. So again, one more time, regulatory sequences, things like promoters and enhancers, they don't actually make any proteins, but they serve as binding regions for proteins that in turn will determine the activity of the genes around those regulatory sequences. So this is a gene, this is a regulatory sequence, and this is a regulatory sequence that determines the activity of this gene. Okay? Depending on what's bonded here and here, that will determine the activity of this thing here. That's what this section's all about. And it's very complicated, and right now there's a massive amount of research going into this because the secrets to this kind of gene regulation will answer stem cell secrets, like how does a stem cell become a heart cell? Can we turn a stem cell into a heart cell and make somebody a new heart whose heart has failed, et cetera, et cetera. So Now we got to talk a little bit about bacteria. Bacteria, in addition to promoters, they also have little regulatory sequences called operators. Operators are little regions on DNA that actually bind to regulatory proteins to shut off genes. So this is a little complicated here, but what we got is 3 is a promoter, 4 is an operator. This is a regulatory protein, and that's RNA polymerase. RNA polymerase would sure like to go down, would like to go this way. These are genes here waiting to be transcribed, but they can't be with this blocker in the way. Okay. Now, this blocker will stay there unless something chemically sticks to the blocker, and that's what this little region right here is for, and changes its shape and pulls it off. If that happens, RNA polymerase can now slide downstream and transcribe and translate the gene. This is a classic example of a gene that is inducible. This gene here, like at number 6, 7, and 8. 6, 7, and 8 represent 3 different genes that would certainly transcribe and translate if they could, but with this blocker in the way, they can't do anything. The blocker is a repressor. So these genes are by default turned off. The repressor is normally there, and the genes are what we say silent. They don't do anything. If this chemical, this is called an inducer. If the inducer comes into the cell, it'll bind to the repressor, change its shape, and pull it off. At that point, RNA polymerase will zip downstream and transcribe and translate these genes. That's a very effective and primitive way that prokaryotes regulate the activity of their genes. That's what this is talking about here. The white crosses here are inducers. They bind to the repressors and they pull them off to allow the RNA polymerase to go downstream. The key here is this system saves a bacteria energy by only producing proteins when there's an appropriate substrate present. As it turns out, these crosses are substrates to an enzyme that will be transcribed from this gene right here. So, if this gene transcribes, an enzyme will end up getting made, and the enzyme will be some goofy shape. It will attach to these little crosses and cut them in half. Cut them in half. Cut them in half. Cut them in half. And that is the little half, that's like lactose. These are like lactose. If you cut them in half, you get glucose and galactose, which then can go off to glycolysis and allow metabolism to take place. So when lactose comes in a cell, it can actually remove the repressor from the gene that actually makes lactase. This is the lactase gene. The cell transcribes and then translates the lactase enzyme. That goes down and breaks down the little crosses. And they in turn are used by metabolism. When all the little crosses are gone, this green repressor will bond back onto the operator and shut the gene back off. Because once all the crosses are gone, there's no longer any need to make lactase. It would be a waste of time to make it. So now that's what this next section is going to go over. And so I'll just briefly talk about it right now. This is the lac operon system. Let's just go right to the diagram here. The lac operon structure is basically what you see in this first picture here. So this lac Z, lac Y, lac A, these are all different enzymes that process the lactose that might get into a cell. The O region here is an operator. This is the binding region for what is called the repressor. The lac operon refers to a cluster of genes that all have a common function. This is the operon. The operator here is used to shut these genes off. Okay. The P is the promoter. That's where normally the RNA polymerase would bind and then slide downstream and transcribe and translate. The cap here is a special binding site for CAP protein. CAP protein is called cyclic AMP activator protein. Some people also call it catabolite activator protein. Either way, CAP is sort of a panic protein in a bacteria. When sugar levels are getting low and the bacteria is facing starvation, it turns to alternate energy sources as a means of surviving. CAP is a way of inducing the production of enzymes that can produce sugar from alternate pathways and alternate substrates. So let's take a look at 4 different states here. In state, let's go to the bottom one here. In the bottom one, we have no repressor. Well, you know what? Best place to start. Sorry, let's just talk about these two. Normally, there is no lactose in a bacteria. That's only in mammals, So if a bacteria is growing in dirt, there's no lactose there. So the bacteria keeps its lactase enzymes switched off. Why? A repressor, the lactase repressor, is normally made. That binds to the operator upstream from the gene, and it switches the operon off. This lac repressor is made by a regulatory gene, not a regulatory sequence, an actual regulatory gene. That gene transcribes and translates the repressor. That binds to the operator on this operon, shuts it off. Now, in these 2 states, in this state, the lac repressor's there. There's no CAP protein. There's no transcription translation. So the XXX means no gene activity in the lac operon. In this state, the panic protein is here. The bacteria is potentially dying, but the repressor is still bound. No lactase enzymes are being made. In this bottom state, lactose must be in the cell. The lactose repressor has been knocked off. This has been knocked off. So there's no square here. There's no cube blocking transcription. So now RNA polymerase can bind here and shoot downstream. The panic button's not pushed. So as a result, you're going to get a low level of lactase production. That will break down the lactose that has come in the cell, which knocked the repressor off in the first place. In this top graphic, what we have here is not only is the repressor gone because lactose entered the cell, but CAP is also there, which means the cell is dying. It doesn't have any sugar. Now RNA polymerase can really fast transcribe. It'll keep binding at the CAP protein region, slide downstream, and express all of these genes as messenger RNA, which will then go to ribosomes and make the proteins, which will break down goofy carbohydrates, which will raise the glucose levels in the cell and keep it from dying. That's the purpose of the lac operon, to keep lactase production inhibited unless lactose enters the cell. If lactose enters the cell but it's in low levels, you get low level of lactose metabolism. If lactose enters the cell and the cell is panic protein dying, high levels of lactase production to produce hopefully high levels of sugar and keep the cell alive. So what you see in the text above here is basically an explanation of what I was just telling you in terms. Inducers, in summary now, inducers are basically like lactose. They bind to a repressor and they turn a gene on. They allow the transcription of the gene. The repressor is normally on the gene and turns it off. That's the lac repressor. That's the little cube down there. In addition, inducers and repressors are rather small molecules. They are proteins manufactured. I'm sorry. Let me reiterate that. Repressors are generally proteins manufactured by transcription and translation, generally. Inducers, on the other hand, in this case, are external chemicals that come in the cell and knock the repressors off. So this is made by the cell. This comes in by diffusion. So inducers are external. Repressors are native and on board. Okay? In addition, regulatory proteins generally inhibit gene expression by blocking transcription. They bind at the operator. Regulatory proteins, though, in general, also can stimulate gene expression by binding to the Tata box to turn it on. Not the Tata box is in humans, but at any rate, to bind to promoters to turn on gene activity. But they can also bind to repressors and knock off repressors. Lactose basically is an inducer for its own enzymes production. So lactose is broken down by lactase when it's made. CAP, catabolite activator protein, which is also cyclic AMP activated, is produced when bacterial glucose levels fall to really low levels, and as a result, CAP then is sort of a panic switch to try to get the bacteria to make alternate sugar to stay alive. Let's see, and let's scroll now to the last thing. We got to talk just briefly about eukaryotes before we finish this topic and we move on to signal transmission. It turns out eukaryotic gene control is a lot more complicated. Eukaryotic gene control is regulated by regulatory genes that make regulatory proteins, regulatory elements, which are can be proteins usually made by regulatory genes that regulate what they do, and transcription factors that directly bind to DNA like leucine zippers and either enhance or inhibit DNA activity. There are specific transcription factors that are now known that bind to specific DNA sequences in the human genome. And some transcription factors actually ramp up gene expression, others cut it off or shut it down. And it's a really complex interaction between transcription factors and regulatory sequences. And genes are all folded over inside your chromosomes. So you can have a gene here regulated by an enhancer sequence that's really far away in terms of linear sequence, but in a chromosome three-dimensionally, they're right next to each other. It makes understanding gene control in the human genome extremely complicated. The level of gene activity, though, and gene regulation in humans can account for phenotypic differences between humans that have the same genes. There's a whole new level of genetics now that we're taking a look at. It's called epigenetics. As it turns out, there can be two reasons why people have, say, blonde hair. Hair color has not been shown to be this way, but like rooster color has been shown to be this way. It turns out that we thought before 1980 that if you had blonde hair, you inherited blonde hair genes. And if you had brunette hair, you inherited brunette hair genes. As it turns out, there's a twist to the story. You could actually have blonde hair genes. Well, actually, you could actually have brunette hair genes and yet be a blonde. And that's because, for reasons that we're only now beginning to understand, your brunette hair genes aren't working. They've been regulated and turned off. That is what is epigenetics. It turns out that we also, in addition to gene regulation in our cells every day, you also inherited some of your gene regulation patterns in the egg and the sperm that made you. That is called epigenetic inheritance. And that is truly one of the most fascinating new fields in genetic research. Understanding regulation of the human genome, especially that human gene activity that's been inherited, the patterns that have been inherited from our parents. So that's it for gene activity. What we're now going to move to is talking about signal transmission between cells.